Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are thrilled to be joined by our guest, Dr. Kevin Murray. Listeners, Kevin is a postdoctoral associate in the Integrative Physiology of Aging Lab at the University of Colorado Boulder. Today, we're excited to talk about Kevin's career path, his research, and the project that led to his receipt of this year's PCC-sponsored Anti-Doping Postdoctoral Award in partnership with the American Physiological Society, or APS. So, Kevin, welcome to the show today. How are you?
I'm great. Thanks for inviting me on. I really appreciate it.
Well, thank you so much for joining us, and we're excited to learn more about you and the work that you do, but let's start by talking about you. So, Kevin, can you maybe give us a more detailed introduction and tell us about yourself?
As you said, I'm a postdoc here at Colorado in Boulder. The laboratory essentially looks at vascular biology in aging and disease, and also one of our interests is how exercise might modulate those in those conditions. So my interest during my PhD shifted towards vascular function, and that has led me to do my postdoctoral work here. And Dr. Seal's lab is one of the preeminent laboratories in the world to study this. So I'm very fortunate to spend the last 4 years here working on this type of work.
Fantastic. And just to maybe take it back to the beginning of your scientific journey, how did you get interested initially in exercise physiology?
I'm an athlete. I've been an athlete my entire life. So naturally, as an athlete, you always want to progress and improve. So back in the mid 2000s, when I was playing basketball in college, the strength and conditioning programs weren't always what they are now. They're amazing. The knowledge is amazing. The ability to improve athletes week in and week out is way better than what it was when I went through. So a lot of us had to do it on our own. So I was going to the library and learning how to improve my eating habits, improve my ability to train in season as well as out of season. So that's really where my interest came from. Was I was always an athlete, but then I really got into the science behind getting better as an athlete. And essentially, I ended up pursuing my master's following that interest and those experiences.
Absolutely. And I think for a lot of people in the anti-doping field in particular, this interest in being an athlete, being in sport, often sparks their interest in the science as well. And you were a basketball player. What really got you interested in basketball specifically?
I grew up in Indiana. So as the saying goes, basketball is basketball in 49 states, but this is Indiana. So I grew up during the 90s with the Pacers, and the Pacers are on a heck of a run right now. They're in the finals and won an incredible game last night in the first game of the finals. So I feel like a little kid again right now, but it was all about basketball growing up in Indiana. That's all we did. That's what we loved to do. It was on TV. The Hoosiers were incredible during that time. So yeah, you just kind of followed what was on TV and got me into basketball. We were always outside playing.
I love that. And we mentioned, of course, that you got this PCC award. How did you initially find out about the Partnership for Clean Competition?
I've always been tangentially aware of PCC, just being an athlete and watching the Olympics and watching the amazing track and swimming athletes. Just off the top of my head, those are individuals that probably get tested a lot, as well as Olympic weightlifting. So I've always been kind of aware of the work and the type of things that like WADA and PCC do to keep those sports clean and fair for everyone. But with the American Physiological Society, we're really fortunate for the organizations like the PCC to provide opportunities for PhD students and postdocs to present their work and receive recognition and awards for it. So I was aware once you guys provided those opportunities, and we're always very fortunate for that.
I think that's wonderful. And Kevin, we mentioned that you're doing remarkable research there in the laboratory, and I'd love to get into some of the details of your research today. So your research has focused on the effects of aging and exercise on vasculature most recently. So can you give us just an overview of some of the changes that occur in our circulatory system with age and also with inactivity?
I'll kind of keep it focused on the vasculature. Essentially, with aging, it is very much in inactivity. With lack of a better term, it's if you don't use it, you lose it. So as you get older and you don't put those incremental strains, whether it's work or exercise, your vasculature, just like anything else, stops working as well with aging. And that's through a variety of different mechanisms. We're mostly interested in the ways that oxidative stress go from just a really important signaling molecule, which is really important for adaptations in response to exercise to improve a variety of different things. But with aging, you have this chronic low level increase of oxidative stress, and we call it excess oxidative stress, that leads to a variety of problems in the vasculature, including reducing nitric oxide bioavailability, so reducing the ability of the artery to dilate in response to a stimulus such as increased blood flow. Or oxidative stress can cause cross-linkings and stiffness in the arteries, leading to increases in stiffness. And that can kind of play on a different variety of things, such as increases in blood pressure or causing end organ damage, such as in the kidney or in the brain or the heart. So everyone ages, so you kind of deal with that in some degree or another, but inactivity can certainly exacerbate that. We look at exercise more so as a necessity. The body in motion stays in motion, it stays healthy, whereas inactivity, the body's not set up to deal with that. So in turn, it has no way to combat that. And in turn, it becomes dysfunctional over time.
Well, that makes sense. And let's talk about then where extracellular vesicles fit into this story. What are extracellular vesicles to start with? And what are the different types that they come in?
Yeah, extracellular vesicles in general are just lipid-bound nanoparticles. And what that means is these are particles that are produced by cells around the body. And they're really kind of an endocrine system. What they can do is the cells, in response to a stress or some certain stimulus, and exercise is certainly like an acute stress, the body can produce these vesicles to then signal and talk around the body. There's been work out there that shows that in exercise, in response to exercise, there's an acute increase in vesicles released from cells into the bloodstream, and they can contain a variety of different cargo, such as microRNAs, proteins, lipids, and so forth. I mean, if you can kind of dream it up, you can think that they have been found to carry even full mitochondria, which is quite amazing.
Oh, wow.
Types is more so size, and it's more so relevant to what they carry. So in general, the society recently has just kind of moved away from categorizing them as exosomes or microvesicles, and more so just calling them extracellular vesicles and just reporting their size. So really, it's more so what they carry, and in response to what stress is what differentiates them.
That makes sense. So then let's put the pieces together here. What role might extracellular vesicles play in modulating arterial function?
Some of the initial insight to this, it's certainly been shown that endothelial cells can release extracellular vesicles, but how they play functionally is a whole nother ballgame. And the work that we've done recently and that we're talking about today is some of the initial research showing that there is a potential role of extracellular vesicles. Now, we don't know where these vesicles are coming from yet. We haven't taken that step to look at maybe the origin of what cell type or what tissue is releasing these to affect the tissue that we're looking at, like the arteries. But in general, we have shown that they can modulate stiffness. And we're in the midst of also looking at how they might modulate the ability of the arteries to dilate as well. So that's kind of a follow-up project that we're starting to initiate. But at least right now, we can state with at least some preliminary insight that they can modulate stiffness of the arteries.
I think that is remarkable. And then let's get into some of the details of the recent study that you presented at the APS Summit earlier this year. So, Kevin, what was the aim of this study?
Sure. Since extracellular vesicles reside in the blood, we were interested in how just bloodborne factors in general. So what we were taking was the serum. So we would draw blood from our participants, spin it down and get serum. And then we were taking this serum and essentially assessing the effect of this serum on modulating arterial stiffness or aortic stiffness with aging. So we had blood from young individuals, blood from older inactive individuals, and then blood from older, what we would kind of call habitually active individuals. And we were seeing if assessing the effect of the blood and whether that modulates differences in stiffness. So stiffness generally increases with aging, and then it can be mitigated with exercise in older adults. And we were seeing if that same in vivo observation can be at least explained by our model through the serum, and whether the serum confers some of those effects.
I think this is remarkable. And I think when we're talking about something like physical activity or even inactivity, it sort of comes along in a continuum. How do you decide, or how do you make the thresholds for the group that is inactive and the group that is active? Like, where's the line in terms of where you might see beneficial or detrimental effects?
The data out there put out by the American Heart Association, ACSM, so the American College of Sports Medicine, generally use this 150 minutes a week. So it's like 30 minutes a day or 25 minutes a day, 6 days a week, or 30 minutes, 5 days a week, however you want to slice it up. And those individuals that meet those criterion for their exercise generally see a robust protective effect. So we kind of use that as our cutoff. And then we use a modified questionnaire where they self-report it. So this initial cross-sectional study that we did was self-reported, and we get a kind of understanding of how active they are and then the type of intensity and the exercise they use. And so we generally use that cutoff as being fully active. And then anybody under that, we kind of consider them as inactive because the protective effects of exercise start to reduce below that threshold.
That makes sense. And so you mentioned you were focused then on the serum and you walked us through a little bit of the methods, but can we go into some more detail about how the study was conducted and perhaps some of the outcomes that you measured?
The assessment that I alluded to before was we see in individuals, we can assess stiffness through carotid femoral pulse wave velocity. And what we can do is assess how fast a pulse pressure after the heartbeat gets to the carotid and the femoral, and that gives us a sense of aortic stiffness. And what we see, as I mentioned before, is that with aging, that value goes up, meaning arteries are getting stiffer, which is not a good thing. And then with exercise, that can be mitigated. They look a lot more like younger individuals. Maybe not fully mitigated, but much lower than their older inactive counterparts. Now, in vivo, we can't necessarily assess serum on the aorta in humans. That is pretty much impossible. What we can do is we use this really cool bioassay that we've produced where we take aortas from young mice. What we do is we dissect the aorta out, we create these small rings, and then we take these rings and we put them in a 96 well plate and we expose them to the participant serum over a 48-hour period. And then following that, we can assess a few things. One is intrinsic stiffness. We have this thing called a pin myograph, and we put these arteries, these aortic rings, on this pin myograph, and we stretch them to get a sense of their elastic modulus, which is a measure of intrinsic stiffness. And we can get a sense then of how much the serum can affect these young aortic rings. And with young participant serum on these rings, we would expect not much of a change in stiffness. But with the older participant, older adult serum, we can see an increase in stiffness, suggesting that the serum is conferring age-related effects. And then with exercise, we see a mitigated effect. Another thing that we can measure that was also in the abstract was levels of oxidative stress. And so we use electron paramagnetic resonance, so a measure of spectroscopy. And we take those same fresh rings and then we can assess them through the spectrophotometer and get a sense of levels of oxidative stress. So not only looking at stiffness, but also a potential mechanism of differences in stiffness that we're seeing.
I think this is fascinating. And just maybe to spell it out for our non-physiologists in the audience today, why is this aortic stiffness in general not a good thing?
The arteries in general are compliant. So what that means is with the cardiac cycle, there's a period of ejection of blood into the arteries, and then there's a period of rest, so systole and diastole. And in order to offset the increases in pressure during the heart pump, which is systole, the arteries have this ability to just kind of have this passive stretching called compliance. And as you get older, with increases in oxidative stress, inflammation, whatever is affecting your arteries, whatever you're dealing with, there's an increase in stiffness, which reduces the ability of the arteries to be compliant and offset those changes in pulse pressures during the cardiac cycle. And those pulse pressures in turn then reach your end organs, and buffering those pulsatile pressures allows for blood flow regulation through like your kidneys or your brain or to the heart. And if those pulse pressures are larger and more intense, reaching those organs, then you end up accruing small amounts of damage over time, leading to kidney disease or dementia or Alzheimer's and heart-related issues. So mitigating stiffness or reducing the onset and progression of stiffness with aging is a really, really important biomedical problem that exercise has shown efficacy to mitigate.
Definitely. And you looked specifically at these aorta rings. Do you anticipate that the effects of the serum that you're seeing on these aorta rings then are maybe systemic, like they would happen the same way throughout the circulatory system? Or is there something special about the aorta in particular?
Generally, you can say yes. I don't know for absolute sure. That's the work we would have to follow up with, whether that's in the cerebrovasculature or renal arteries or whatnot. But generally, if you see changes in the macrovasculature, which we consider the aorta the largest artery in the body, you generally see increases or dysfunction in the microvasculature. So just based on some of the literature out there, I would suggest yes. What you're seeing in the aorta is probably going to be transduced throughout the vasculature.
That makes sense. And you mentioned some of the findings. Let's then talk about the bigger picture, Kevin. What are the implications of these findings, or where do you go from here?
we're really excited about the findings because anything you see in vivo and then you can recapitulate the phenotype ex vivo or in some mechanistic modeling is really exciting, especially when this is about as close as we're going to get and doing this in humans. So we consider this model a very advanced and translational model that gives us a sense of what's going on in the human body, even though we can't necessarily do that. So we are writing grants up. We're trying to get this work funded to get a larger study. This was done in a small amount of individuals just to get a sense of preliminary and pilot understanding. So we've put in some NIH grants recently that are hoping to be reviewed and scored here soon to see where we stand and what we need to do to change our approach in order to get this work funded. But that would be to do cross-sectional analysis, but then we would like to do some interventional analysis of pre versus post and seeing if we can modulate that versus just looking at a cross-sectional slice of people who do exercise, people who don't exercise. This gives us a sense of like, yes, in fact, we can produce extreme causality if we can give individuals an exercise intervention and then see the serum play a role pre, post, and exercise intervention, bringing, like I said before, causality to the whole piece and work.
Absolutely. And then to make the connection with sport and anti-doping, I know in most cases, extracellular vesicle therapies are not currently prohibited in sport, but they're sort of becoming a hot topic in the area. So based on your work and maybe research from others as well, Kevin, what do we know and what gaps remain in determining just the kinds of therapeutic effects that these extracellular vesicle therapies, things that could be put into the serum, into circulation, could have, and whether there should be concerns surrounding potential use for performance enhancement?
The therapies are certainly advancing and they're showing efficacy and potency, definitely in preclinical models. And there are some advancements at the clinical level. Obviously, they're not widespread. They're not really implemented all that well. So the gaps remain. A lot of it has to do with safety. A lot of these types of things, yes, they may show efficacy, but there can be a multitude of off-target effects. They're unknown. So you might be wanting to improve skeletal muscle function or artery function, but the vesicles are not going to just choose based on where you want them to go and be able to make sure that they are received in the tissue of importance, the precision. All of that has to be worked out before this can be considered safe. And it really does come down to safety. A lot of it is just because it shows efficacy doesn't mean it's great. So I think it's pretty well known that the vesicles play a role. The vesicles are potent. I think the vesicles will do something in the future and will be a player in the future, but everything needs to come down and boil down to safety. And without that, then a therapy is really not very useful.
Definitely. And I'd like to dig into this idea of safety and risk. You mentioned specifically off-target effects. Could you maybe give some examples of what are some of the potential risks with extracellular vesicles or even just other biologic therapies?
In general, a certain molecule could be very beneficial for one cell type, just to stay general, and not so beneficial for another cell type. So yes, you may see, let's say you're trying to increase skeletal muscle strength or just oxidative capacity or whatever it might be, reductions of fatigue. Things like that for the muscle might be very healthy, but for another cell type could cause uncontrolled growth, or it could hamper the immune system, or it could cause clots, or just a plethora of things that you don't know until you know. So just the fact that extracellular vesicles, as far as we know, are not necessarily precise in general. They're released and then they're taken up by cells when they come to and dock. It is exciting, but it leads to a lot of safety and dangerous questions of, if you give something for recovery, is that going to improve fatigue and resistance to fatigue in the muscle? But what is it going to do for your kidney cells or your liver cells? These things that a lot of times you don't even think about related to exercise, but in the longevity and health of an individual, they're incredibly important.
Absolutely. It sounds like a lot of concerns surrounding maybe the lack of specificity of the treatment or the inability to sort of do this precision targeting with the extracellular vesicle therapy. And just, it sounds like there are a lot of questions that remain unanswered. And I think it's important to continue doing research in this area. And of course, we mentioned recently you received the PCC-sponsored Postdoctoral Research Award in collaboration with APS to support you presenting some of this research. And you presented results from your work at the annual APS meeting. I'd love to talk about this experience. What was it like for you to attend the meeting and share your work?
It's always fun to present your work, and even more so present your work around individuals who have similar interests. So we got to present, it was kind of like a data blitz, and you got a couple minutes to go up there and you had a couple slides and talked about your work. And it's kind of almost served as like a marketing for people to come and talk to you at your poster. And I had a lot of excitement at the poster. I had some great questions about what are the next steps, some of the things we've talked about here, implications, people who wanted to get involved with the work. So it was really exciting. So the opportunities to talk to individuals who you normally wouldn't necessarily get to talk to, but maybe aware of the work that they do or the awareness of that they work in that field. And then some of them stop by your poster and talk to you about your work was pretty awesome. So just the ability to go to that society meeting, get recognition for your work, because you always think, one, your work is pretty cool, or others to also think that your work is pretty cool is the ultimate, especially in science. And then have people come up and be excited and some big players in the field talk to you about your work. It was exciting.
Well, it sounds like a fantastic experience, and it sounds like the research you presented was well received by the audience there. And I'd love to talk about just what's next for you. I know these early career portions of your journey in science involve some transitions. So what are the next steps for you in your work, Kevin?
So the biggest thing is in the acute is to try to get this work funded. That would be very helpful to then take that on research in general. If you can market yourself, it's much easier to do so when you have grants that support your work and showing that you can go out and get extramural funding. Awards are nice, manuscripts are nice, but funding is a really big, important piece to that. So the grants that we've put in recently related to this work will help with those transitions of finding a position at a university or somewhere else to continue on with this work or in this area of research. So yeah, really being a senior postdoc, that's really where a lot of my thought goes is how can I package my work to become competitive and marketable to other places to find a position and then really kick off my own lab to take this in a direction I think is really important.
Absolutely. And I think the work that you're doing is relevant to a lot of different fields. We mentioned, of course, anti-doping science, but also there are clinical applications to your work as well. And maybe just taking a step back, can you think about and tell us a little bit about the important contributions that people with an exercise physiology background can make in the space of anti-doping science?
I think where we can make huge headway is some of the initial just pilot findings of just the involvement, functionality. I think the abstract in this work represents that in general. We have some of the freedom to kind of go after ideas that other places might not necessarily get to as an academic scientist. So we kind of have this freedom to explore different things and do different things that maybe industry or companies that are not necessarily able to because they have a certain mission and their work has to stay in that lane. So academic exercise physiologists, as we mentioned earlier, we're athletes. And so we see how this work might be relevant. And then we can take that and use some of the models we've created and create some really initial basic and foundational understanding of how, in this case, extracellular vesicles may play a role in aging and how exercise may modulate vesicles to mitigate stiffness with aging. So just using this as an example, I think basic foundational understandings is where we can really make our biggest contributions, especially where exercise physiologists, where a lot of the stuff used to be done just in whole body and kind of just understanding of processes, like macro processes. Now, a lot of the exercise physiologists, we consider ourselves almost like molecular biochemists interested in exercise physiology because everything is now about, okay, so why does this happen? What's going on at the molecular level? So a lot of us with that freedom, we can really take it in a direction that can give us some clear and strong resolution at the micro and molecular level. I think that's where we can make our biggest contributions.
Absolutely. I couldn't agree more. I think this idea, particularly thinking about extracellular vesicles, a lot of the foundational work still remains to be done to really understand the kinds of impacts that these kinds of therapies could have, to know how relevant they are for anti-doping and in what circumstances and what steps need to be taken to ensure clean sport in light of them. So we appreciate the contributions that you and your colleagues are making in the space. But I know it's not easy being an early career scientist, really in any field. So, Kevin, what are some of the biggest challenges that you've faced as an early career scientist?
Aside from funding, I think just carving out your niche. A lot of people are doing some amazing science, and to find a place where you sit and you can contribute not only just in the next 2 to 5 years, but where you can make a career out of it takes a lot of thoughtfulness and effort and mistakes. Learning to be self-aware, learning to be able to start over, learning to be able to be resilient and perseverant, I think is incredibly important. That's something that you only learn by failing. And a lot of times we're perfectionists to some degree, and that's difficult to accept. So being willing to fail and willing to go out and try new things. A lot of times, like, we show you the data and this award is a product of data that worked. The majority of the time, that's not the case. So just being able to go back to the drawing board and trying again. I think that's more indicative of success as an early career scientist than maybe where you trained or what kind of grades or how smart you are. It's more so like who's willing to get kicked off the horse and get back on again and try again in a different way. Those are some of the biggest challenges that we face early on, is that if you fail, which is going to happen, to just be able to continue to try again, especially with how difficult funding is and how difficult it is to get funded. It really is a success story when you do get that grant that comes through and your work gets funded.
Absolutely. I think a lot of science is how you respond to the challenges you face. And I think that can help determine your success. And we mentioned that this APS PCC project that you presented was a success. Was there a challenge or a failure, an example of something you struggled with in this particular project that you could tell us about?
We've been dabbling in serum-related phenotypes for quite some time. We have different models, cells, and here now we have this mouse artery model, but we had never really isolated extracellular vesicles, and we were really pretty green, naive to the field just a few years ago. So really trying to learn, one, how to find a method to isolate extracellular vesicles cleanly and in a manner that was feasible to us with access to certain types of instruments. It probably took, of the 3 or 4 years that this has been going, more than half of the journey. Once we kind of got that and we were consistent with that, and then we were able to start taking these vesicles and reliably putting these on the arteries, and then in turn keeping the arteries alive, and then assessing their stiffness or their oxidative stress. Once we got going with that, that came pretty quickly in about 6 to 8 months. But the prior years was figuring out how to isolate vesicles and do so in a manner that they remain functional and that we didn't kill the arteries and just the concentrations and everything. So really, it was a constant trial and error for years. And then now this is the result after 3 or 4 years worth of trying and trying again. So,
well, I think that is remarkable, Kevin. I think it's so helpful to hear the context of what it's like to see a project like this through to the end. And we talked about the challenges, but we like giving scientists the opportunity to dream big on the show. I know oftentimes you're limited by things like funding and resources and time and all these little details, right? If we took away all of the barriers that normally hold you back, is there a particular problem in anti-doping that you would most want to solve, Kevin?
Generally, testing for safety is obviously one of the biggest reasons anti-doping is out there. Safety, as well as just keeping the playing field fair. I think somehow trying to figure out a way to stay ahead of the types of technologies, we're always behind, right? So something is introduced and then the testing, the safety and everything comes after the fact, because a lot of times as scientists, we think of how our technologies are going to be used to help people and health, and we don't necessarily think about how they might be used aside from that. And I think finding ways or getting in a vein or artery of thought, without having too big of a pun, trying to stay ahead of the technologies out there, maybe sitting down and thinking about ethically what the type of technologies we're producing and how they might be used inappropriately in the terms of like anti-doping or doping and getting an unfair advantage or putting athletes in less safe situations. So I think somehow the mindset around just staying ahead of the curve and sitting down and thinking ethically about what we're doing, what we're bringing to the table with these new awesome technologies that are meant to help people, how they might be used inappropriately. And I think that is something that if a lot of effort is put into that, we could keep athletes and just individuals in general safer and be ahead of it versus having an athlete get hurt or somebody hospitalized or hurt. Then we find out that that's what's going on. So I think the preventative aspect of that would be a really all resources imaginable. I think that would be the biggest bang for our buck.
Definitely. I think you brought up some really great points there. Just this idea of thinking through the potential safeguards and guardrails. What can we do to ensure that new technology, new therapeutics, new drugs that are being developed aren't misused? What can we do to ensure that they're being used safely and maybe only getting into the hands of the people who really should be using them? I think would be remarkable.
I think that's probably $1 million question. Humans are human. People are going to always try to get an advantage. And I don't know if there is an easy answer for that. I think if we knew the answer to that, the problem wouldn't necessarily exist.
Right.
As scientists, before we put things out there, I think we really need to think everything through. We're really excited because we're always trying to find a discovery and we're trying to get it out there and published. And a lot of times you can get a patent on it and potentially receive some compensation in turn for that. So as academic scientists, we don't make necessarily a lot of money. So I think a lot of times they're not doing it on purpose, but they go out there and they try to make a name for themselves. And that's what we do. Our job is our ideas, our products are our ideas. And I think sometimes just taking that extra step and thinking about that before we commercialize certain things or start implementing certain things, just sit down and think about how this needs to stay out of people's hands and who needs to control the substance or the technology to make sure that it is only put in the hands. So I think it really comes down to us, the people who make the ideas, and making sure that before we put things out there, especially if it can be used quite quickly at a benchtop at home or something like that, to make sure that those types of things are regulated before they're put out there for the public to know about it and use it.
Oh, that makes sense. Well, Kevin, we appreciate you sharing your ideas and your insights with all of us today. If you've grabbed the attention of our listeners and they want to know more about work in this area, what is the best way for them to learn more about you and your research?
Probably the best place is, I don't know if most people know about this, but people are involved in research. There's a Google area that they have Google Scholar citations. So as a researcher, the Google search engine allows collection of all of our articles, whether they're abstracts or manuscripts, and kind of puts it all in one location. So I think that's probably the best place with the things that I've put out there in that have at least been either peer reviewed by conferences or peer reviewed in journals to get a sense of what else I'm involved with. So I would say my Google Scholar citations page would be the best place for that.
Excellent. Well, listeners, definitely take a moment to check out Kevin's Google Scholar Citations page, learn more about the amazing work that he's been doing. And Kevin, it's been such a pleasure to have you on the show. We appreciate you taking the time to share more about your career and your work today.
Well, thank you very much. It was a pleasure and a surprise for the invitation on here, and I really enjoyed it.
It was wonderful to chat with you, listeners. Always great to have you here with us as well. If you haven't already, it would be wonderful if you could rate our podcast and leave a review on your favorite podcast platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.